It's 9 o'clock and we're going to get started here. My name is Scott Plevy. I'm the course director. I'm a gastroenterologist from the University of North Carolina School of Medicine in Chapel Hill. We're looking forward to a great day of lectures from really people who are experts in all of their respective fields across different disciplines in clinical immunology, which is the theme of this course. This is the fourth year that we've had this course at the Focus meeting. As a reminder, this is a CME course. You should have all received your pretest prior to this by email. After the course, you'll receive a post-test as well as CME certification information via email as well. The course learning objectives are shown on this slide here. You should be able, after completion of this course, to identify scientific basis for choice of immunologically related therapeutic targets in various diseases. I won't read the diseases. You'll hear about them today. Explain the rationale and mechanism underlying major pharmacologic approaches for interventional immunology in current practice. Evaluate the track record of different therapeutic approaches in different specialties. Recognize the cross-disciplinary lessons that can be learned from the clinical experience with specific interventions, including pharmacologic issues, limitations in altering disease progression and complications due to immunosuppression, and analyze how a physician should decide to use and choose these therapies. Without further ado then, I would like to introduce our first speaker for the morning, and it is Amit Bar-Or from the Montreal Neurological Institute, who is going to talk to us about the immunologic basis of multiple sclerosis and new therapeutic targets. Forward, backwards, laser.
Thanks, Scott. Okay, good morning, everybody. Thank you, Scott. It's a delight to be here. This is not the correct talk, actually. Many thanks for the invite. I've been given the challenging task to try to both update you on some of the emerging themes in the thinking about multiple sclerosis pathophysiology, and in that context to tell you a little bit about some of the emerging therapies. Your challenge is to try to bear with me over the next 40 minutes or so as I do this. The first disclosure that I have is that I apologize you will not find the notes from my talk in your presentation, and this is the little culprit responsible for this. After a bit of a rocky neonatal ICU course after being born at 29 weeks, this is Leora. We became real parents just a week ago, and so we're enjoying the sleep deprivation, but this is also the reason why I wasn't able to provide the slides in advance. My shift is the 4 a.m. shift, so I prepared the talk this morning at 4 a.m. Gail assures me that these will be available online at the Focus site within 24 hours. So apologies again. The other is to thank Scott for the flexibility and Kevan for switching with me, and apologize that I'll have to leave after the talk to give another one, as well be away in the afternoon session. The more mundane disclosures, but important, are listed here, and my interactions with pharma have included pharmaceutical companies that are responsible for some of the agents I will speak about, and of course the last bullet point there is that by definition I'll be talking about some investigational as well as off-label drugs. So as an outline, I'd like to start with the first dozen slides or so with a brief overview of the immunologic basis of multiple sclerosis and really underscore what is a challenge for us in the field of MS. We're dealing with multiple compartments, and we're dealing with an evolution within these compartments of multiple biologies over time, which of course challenges us with respect to targeting this disease effectively from a therapeutic perspective. We'll talk briefly about the presumed modes of action of established therapies and spend the bulk of the talk discussing some of the emerging therapies. There are literally dozens in the MS field, and I'll have to focus here on ones that are fairly advanced in clinical trials, phase 3 and recently approved. These are the ones that the community is now dealing with and trying to figure out how to build into our armamentarium. And the subtext to this presentation is the bottom bullet there, which is we should think about how these experimental therapies, when they jiggle the pathophysiology of a complex human disease, may teach us something about the human disease itself. And so MS, like in other human autoimmune diseases, is generally viewed as a disease in which autoimmunity, which of course is normal, we all have autoreactive B cells and T cells, but insufficiently regulated in those individuals in whom autoimmune disease emerges. And it is thought that this is on the basis of multiple genes interacting with environmental exposures over time, leading to these states of dysregulation. And in MS now, the whole genome studies, the HAP maps and so on, have really suggested that the multiple genes that have emerged as implicated in susceptibility, each, by the way, contributing only a tiny bit to susceptibility, but they essentially converge to what we consider to be immune-related genes, so supporting the view that MS is primarily an immune-mediated disease. There's still a lot to be discovered in terms of the gene-environment interactions, and the targeting tends to be of established MS, where we're already dealing with abnormal states of regulation that we're trying to correct. The typical three-compartment model, which I expect is common to many autoimmune diseases, in MS has as the target tissue the central nervous system, the brain and spinal cord, separated by the famous blood-brain barrier, which in fact is a very dynamic organ in and of itself, and that barrier separates the CNS from the peripheral immune system, where we believe there are important aspects of peripheral immune activation, and then a cascade of interactions of activated immune cells that more efficiently adhere, become attracted to, and actively invade across the barriers to become reactivated within the target organ, where they bring to bear their effects on the glial and neural elements of the CNS. And this, though simplified, nonetheless provides us with points where we can consider as targets of current and future emerging therapies. Now, that model seems to be consistent with what we know pathologically in MS. When you look here at this coronal section, these areas of brown-yellow discoloration underneath the cortex within the white matter and periventricularly in the white matter are the classical perivascular inflammatory lesions, here staining for a pan-T-cell receptor antibody, so each black circle is a T-cell, and this is the oblique section with the lumen of the vessel here, very dense perivascular inflammation at the sites of segmental demyelination and axonal transection, as shown here. And so this is in keeping with that little cartoon model that I had pointed to you. However, there are some important conundrums in the MS field that I'd like to share with you that contribute to our evolving thinking about mechanisms of injury. At the top, we see the typical relapsing remitting onset of the disease, and what has been described to occur in most patients, at some point they transition, no longer having attacks, but continue to relentlessly progress. At the bottom, superimposed on the same timelines, are a couple of brain imaging parameters that you might track patients' disease in vivo over time. The green represents the total volume of lesions as assessed by, for instance, T2 hyperintense lesions on the typical clinical brain scans. You can see increasing fairly rapidly early in the disease, but much less so later on, yet patients continue to get worse. So this is a dissociation between what we see clinically or by imaging and what we think is happening under the surface. The other is represented by these little black arrows. Each would represent a time point where you would capture gadolinium-enhancing lesions in the brains of patients, and those are taken to represent areas where there's local breach of the integrity of the blood-brain barrier, where you might have the gadolinium contrast collecting and staying in the tissue and then showing an enhancing lesion. You see a higher density or higher frequency of these events early in the disease and fewer later, again at a time that patients are nonetheless getting progressively worse. So we have to accept that underlying the injury in MS are at least two distinct biologies, if you like, the blue and the red, which people have referred to at times as inflammatory versus degenerative. My view, and I think what is emerging now, is that the blue can be considered one where there's a peripherally initiated process, or at least an important aspect to peripheral immune activation, and these waves of perivascular inflammation contribute with the clinical correlate of the relapsing remitting disease. So the relapsing remitting biology would be the blue biology. The red, however, represents CNS compartmentalized processes, probably plural, and I'll try to convince you of that in the next few slides, and involves likely local CNS compartmentalized inflammation and some aspects of actual degeneration of tissue, meaning if you took away the inflammation, there would nonetheless be dropout of local tissue over time. So this is what we're now thinking of as the complex timeline of MS and the multiple biologies, and the next few slides will underscore this theme. We now appreciate that MS is no longer a disease just of white matter, but the gray matter, the cortex, is also rather heavily involved. And this shows you several types of demyelinating lesions, this one extending from around the vessel within the white matter into the cortex, this one within the cortex, normal cortical myelin, and here around the vessel a punched out demyelinated lesion within the cortex, and this is the more dense normal white matter myelin versus the normal cortical myelin. This lesion, however, is like a slab of demyelination underneath the meninges and the pia. So this has been referred to as the subpial demyelinating lesion of the cortex, and it does not respect the distribution of a blood vessel. We cannot invoke perivascular inflammation as the mechanism of injury, and as shown here in this reddish-brown color, whatever this color is, this represents topographically the surface area in this section, but you can imagine the volume involved in this subpial cortical demyelination in this particular individual with fairly advanced secondary progressive MS. And it is thought that this type of injury, and what is a much more diffuse injury in the CNS of patients than the multiple perivascular focal lesions, is likely to represent the biology that underlines the progressive decline of patients over time. So that means that when we think of therapeutics, we need to target these multiple compartments and subcompartments, indeed, even within the CNS, as they evolve over time. And this is a somewhat more complex cartoon that just summarizes that within the CNS, we have the typical perivascular lesions, we have this cortical injury, there's now description, similarly to what has been described in the context of target organs of other chronic inflammatory diseases, collections of immune cells described in MS sitting in the pia and meninges, some of which appear to recapitulate follicle-like structures. And the question that is attractive to speculate on is, how might they be involved in contributing to the type of cortical injury that one now appreciates? And then we need to think of how our therapies, which are generally administered in the periphery, might impact the peripheral immune responses, and will they or will they not translate into changing the face of the disease within the central nervous system. So these are our challenges, and that's the brief, rather limited update into our understanding of MS. Now, these circled perivascular lesions are the ones that we have been targeting over the years and becoming increasingly effective at doing, and really the talk will focus on these, but to underscore the point that we have a lot to learn about the neurobiology of the disease, about the immunology and the inflammation within the CNS, and we're only kind of pushing the envelope now on understanding these underlying biologies and how to develop therapies that reach the CNS and beneficially impact these biologies. So most of our talk will focus on targeting the perivascular invading immune cells in the periphery, but you'll already see that some of the agents we'll be talking on that are emerging seem to have an impact on the neurobiology of the disease, which is one of the exciting new emerging themes. Now, much of our thinking in terms of peripheral immune activation impacting the target organ in MS comes from animal modeling. The most commonly used EAE, experimental autoimmune encephalomyelitis, which is induced in an animal when you inject it in its periphery with a piece of the central nervous system. You activate in the periphery immune cells directed to this target antigen. The cells then get into the CNS, identify the target, and the animal comes down with this paralytic illness. It's been very helpful and continues to be in teaching us principles of peripheral immune activation and targeting of the CNS, but EAE is not multiple sclerosis for a number of reasons. It's induced, whereas, of course, in humans we don't think we actively induce people to get MS. Humans, of course, live in much dirtier environments than the animals in their facilities, and we, of course, are much more genetically heterogeneous. It may be that the animal modeling is better for the blue biology, but certainly we do not have to date animal models that are good models of what happens in terms of the neurobiological aspects of the disease and the disease underlying progressive decline in our patients. Now, in MS, and again, largely driven by animal models, but still we think this is relevant to patients with a human disease, life used to be relatively simple when the T-cell world, which has been thought to be the main mediator of disease in MS as well, was divided into Th1 and Th2. I think all of you will appreciate, and you'll see many of these increasingly complex slides, is that we're dealing with multiple effectors, we're dealing with multiple regulators, and to keep a long evolving story very short, we do think that the bad guys in MS, certainly in EAE, but also in MS, are the Th1 and Th17 T cells. Also CD8 T cells appear to play an important role. And then on the regulatory side, there are indeed multiple regulatory cells, and defects in several of these have been identified in MS as well. Now, of course, we recognize that it's not just the CD4 T cells. There are multiple other effector cells that have been implicated as important mediators of injury in MS. There are multiple different regulators that have been implicated as being deficient in the context of MS. And indeed, there are many cells, of course, and other factors that contribute to the decision-making T cells make in terms of differentiation. So there's no escaping the complexity of the immune system in MS as well, and I think this will be a theme that will underlie many of the talks that will come after mine. So back to our simplified immune model of multiple sclerosis, which focuses mainly on this context of peripheral activation and subsequent invasion. What can we think of in terms of, or what can we discuss in the context of some of the emerging therapies? Now, in MS, patients have been exposed to the full gamut, ranging from antigen-specific attempts at modulating those few cells that are thought to be the putative bad guys, all the way through to completely ablating patients with high-dose immunoablation, chemotherapy, and then reconstituting their immune system, typically with autologous CD34 positive hematopoietic stem cells. And very simplistically, one can consider changing the profile of these drugs as you move through the spectrum from those that might be viewed as rather safe because they only affect those particular bad guys without impacting the rest of the immune system, but may not be as effective, to drugs that have been showing increased efficacy, but of course at a price. Now, our approved immune modulators are sitting here: interferon betas, letermovir acetate, natalizumab, mitoxantrone, a chemotherapeutic, also approved, belongs to the immune suppressive family. In brackets are medications that have not been approved. So really these represent the approved therapies. The antigen-specific therapies, none of course approved, but I'll put in brackets. We may want to discuss these at the end if we have time. They have certainly taught us some very important things about the capacity to modulate antigen-specific responses in the periphery of MS patients and change the face of the disease within the CNS. So that's a sort of proof of principle, but we've also run into problems where, for instance, the altered peptide ligand of MBP actually induced new brain lesions rather than diminished new brain lesions. So we need to learn how to safely bring this type of treatment to bear, because it does continue to have certain attractions. Fingolimod, or Gilenya, I'll talk in some detail about, has just recently been approved in the US and Canada for the treatment of relapsing remitting MS. And if we have time at the end, we'll talk a little bit about selective depleting therapies, including anti-CD20, B-cell directed, and anti-CD52, a much broader depleting agent. As far as a very brief, really brief overview of the presumed mechanism of action of the already established therapies. These are therapies that have been in the MS world for approaching a decade and a half, 2 decades. Interferon beta and letermovir acetate, they're all injectables. Interferon betas work through targeting interferon receptors on multiple different cells, not just immune cells, but in context of MS-relevant immune pathophysiology, they're thought to decrease T-cell activation, perhaps change profiles of Th1 to Th2, but certainly impact the interaction with the barrier by decreasing substantially the capacity of these cells to upregulate adhesion molecules, and by directly impacting their capacity to produce the matrix proteases that need to actively break down the barrier for cells to invade. So that translates into less inflammation within the CNS. Letermovir acetate, a very interesting copolymer of 4 amino acids, GLANT, works, we think, as an antigen, which modulates the profile of antigen-presenting cells more to a type 2 APC, modulates populations of T cells more to a Th2 rather than a Th1 or Th17 profile, and probably induces regulatory cells as well. So these changed populations contribute to less inflammation in the periphery, and the thought is that these cells, because they're activated as Th2 cells, have the machinery to get across the barrier, but there, through bystander suppression presumably, decrease inflammation. So that's really a very short overview of how we think these very established therapies work. Natalizumab is a very interesting targeted molecule, which is an antibody against VLA4, one of the important adhesion molecules when it comes to immune cell blood-brain barrier interactions, and has been very effective at decreasing MS relapses. However, we now know that somewhat more than 1 in 1,000 patients, so infrequent, but still important, because about 1 in 800 patients after a year of continuous therapy will come down with a very bad brain infection, which is because of reactivation of the JC virus, leading to what's called PML, which is a nasty demyelinating disease of the CNS caused by Tysabri. And so one of our challenges in the field, and I think another theme that will run through many of our diseases, relates to how to try to stratify patients with respect to risk. Can we identify who's most likely to benefit, who's least likely to benefit, and if we knew who the 1 in 1,000 or so patients would be who would come down with PML, of course the other 999 would be on this agent. So Tysabri, a very effective agent, but with one particular nasty side effect, which is limiting its broader application today. Let's now segue into some of the emerging therapies. And again, this is going to be focusing on a few, particularly on some of the oral therapies, because this, after years of only injectables, is one of the most important advances in the MS field, and then to drugs that have pretty strong and in some cases surprising efficacy, but may not be ideal for long-term use. Nonetheless, they may teach us important themes about the MS disease process and how to target it better. So Gilenya, also known as fingolimod or FTY-720, has been recently approved for the treatment of MS, relapsing remitting MS. It has been borrowed, if you like, from the transplant immunology field, so some of you may be familiar with it. It is generated as a synthetic analog from sort of learning from fungi, and in fact targets sphingolipid metabolism. So this is the FTY agent. This is sphingosine, which is important in the metabolic pathway of sphingomyelin through to the palmitoylated ethanolamine phosphate forms. And what you see here is that FTY-720, just as sphingosine, when they get phosphorylated, either the endogenous ligand or the FTY, which is ingested and phosphorylated, acts on sphingosine 1-phosphate receptors. There are several of them in humans. They are present on many, many cells, both of the immune system shown down in the right, but also within the nervous system. And this is a very lipophilic agent, so it does get into the CNS, and I'll get back to that in a moment. In the context of the activity on the immune side, this is a very interesting agent, and still a lot is being learned about it. What it does is, quite reliably in patients, decreases lymphocyte counts in the periphery because the sphingosine 1-phosphate receptor is responsible for the capacity of immune cells, particularly CCR7 positive immune cells that circulate through the lymphatics. This is responsible for their egress from the lymphatic tissue. And the idea is that the FTY in its phosphorylated form will bind to the S1P receptor on these immune cells, downregulates the receptor so that when these cells enter the lymph node, they no longer have the receptor on their surface to be able to sense the endogenous S1P ligand and then egress. So they get stuck in the lymphatic tissue. And this sequestration has been thought to be a main relevant mechanism of action for decreasing the lymphocytes in the circulation, including presumably the bad guy cells, so that less can access the central nervous system. It turns out that work, this is by Trina Johnson with Jack and Tell from our lab, looking at some of the functional consequences, so not just changing the profile and the numbers of immune cells in the periphery, but if we take CD8 cells that are less impacted than CD4 cells and actually take them out of the periphery of patients on FTY compared to untreated patients who get FTY in vitro only as opposed to on therapy, and looking then at the similar CD8 cells taken out from a normal healthy donor, migrating them in this migration assay across a barrier, you can see a fairly profound impact on this functional property. And this is just one example of how the cells that do remain in the circulation may not harbor the exact same biologies as their phenotypic counterparts prior to the intervention. So there's a profound change in the profile, but we also think to some extent, at least, on the function of the cells. And from a therapeutic standpoint, this is looking at annualized relapse rates in the relapsing remitting population and looking at 2 doses of fingolimod against active treatment with one of the interferon 1 betas, the weekly injected. And you can see that better than one of the standard therapies, the fingolimod at both these doses is quite effective at decreasing, although not eliminating, annual relapses. This comes at a price. It is not a very high price for people dealing with other diseases. The MS world and neurologists tend to be a little conservative, so we tend to look at these red flags and get a little concerned about what is a chronic lifelong disease. But you can see that as far as percent or frequencies shown in the brackets, percentages of any of these complications are not high, but altogether maybe 3 to 5% of patients on this drug over time will run into some problems. There have been some infections, including herpetic infections, 2 of which were fatal. Patients who are not showing immunity to VZV need to get immunized, and they should avoid steroids, which was one of the contexts in which a patient died from disseminated herpes. Interestingly, I'd alluded to this. This is just one example from Veronica Miron and Jack Antel's lab showing you that in vitro, FTY-720, when added and shown here in a dose response, can protect and save oligodendrocyte precursor cells, OPCs, the cells that are thought to be relevant and critical for remyelination within the injured central nervous system. And indeed, this is one of those nasty cartoons, one that I apologize we contributed to recently in a review in Neurology, but showing you again the three compartments and how one now believes that FTY has an impact, as I had mentioned in some detail, on the immune response profile in the periphery, seems to actually have an impact directly on the barrier, which I have not shown you, and then within the CNS, there are now 7 or 8 papers, both in animal models and some early suggestion in humans, that this drug gets into the CNS and may have an important impact on the neurobiology of the disease, representing now a dual action agent, which of course would be particularly attractive in the context of these patients. The next drug that very likely will get approved over the next year or so in the context of relapsing remitting MS is again a borrowed drug. This shows you fumaric acid, Fumiderm, which is a fumarate treatment of psoriasis. So it was borrowed from this, and in fact BG12, which is the dimethyl fumarate form of fumaric acid, was shown in the animal model EAE to have a relatively mild benefit in this particular model, but in the face of a relatively modest effect on the peripheral immunology, seemed to have a fairly good effect in terms of preserving axonal density and decreasing demyelination. And interestingly, Ralph Gold's group, to pick up on the same theme I had mentioned in the context of FTY, has shown elegantly in some animal models that fumarate, basically using the BG12 in an animal model of CNS inflammation, seems to have neuroprotective capacities and implicated the NRF2 antioxidant pathway. So another drug where there is hope that this will work both on the immunology and the neurobiology of the disease. And in fact, good news just released recently at the American Academy is that there was a fairly substantial reduction in annualized relapse rates and a fairly important impact on new lesions on brain MRI, both the T2 lesions I mentioned and these gadolinium enhancing lesions that are important for patients. This drug is easy to tolerate with some GI side effects that limit it in very few patients, and will be a welcome addition to the MS armamentarium. Another oral therapy is teriflunomide. Teriflunomide is the active metabolite of leflunomide, which some of you will recognize, Arava, which has been approved now for some years in rheumatoid arthritis and psoriatic arthritis. It is a non-competitive, reversible, and selective inhibitor of this particular mitochondrial enzyme. And it's interesting because this is a key enzyme in the de novo pyrimidine pathway of pyrimidine synthesis, but does not affect the alternate, or what's been called the salvage pathway. And so pyrimidine, while it is very important in many biological processes, and if you completely prevented its synthesis, you would certainly run into big problems, because of this relative selective targeting, it seems to result in very little toxicity, certainly no cytotoxicity to immune cells. It is thought to work by immune modulation of immune cells, including T and B cells, although the MOA as relevant to MS is really not fully worked out, and there are studies that are now pursuing it. It does not seem to have much of a penetration across the blood-brain barrier. It has been shown to have pretty promising results in a phase 3, as well as supporting phase 2 data already published. The phase 3, fairly large study, had identified significant reductions again in these primary outcomes of frequency of relapses and multiple MRI and clinical secondary outcome measures. It was quite well tolerated. It has some liver toxicity, which is transient. You do some monitoring, you catch it, you stop the drug, and the liver enzymes tend to come down quite quickly. So again, what will likely be a relatively well-tolerated and relatively safe drug, oral, which is going to be welcome, and we're waiting for the final phase 3 study results to come out to get a sense of how it might fit into the armamentarium. And the final oral therapy to touch on is laquinimod. This is a quinoline metabolite, and again developed as a follow-up to a drug called linamide that had been tried in multiple sclerosis, looked actually to be quite effective, but resulted in several complications, including MIs and pericarditis. And the reasons for this are not entirely clear, but they were able to develop a beagle animal model of serositis, including pericarditis and pleuritis, and then used an in vivo structure activity relationship, or SAR screening, to take this drug, the linamide, and to modify it in many different ways to generate a panel of agents that were then tried both for efficacy in EAE as well as for the potential toxicity in these beagles and came up with laquinimod, which worked better in EAE than linamide and had much less of an impact, in fact none seen in the beagle model with respect to this particular side effect. So this has been introduced to patients. The primary outcome of the phase 3 study shows a relatively modest reduction in the annualized relapse rate in the face of what is a fairly substantial reduction in EDSS progression. That doesn't look impressive, 33%, but in fact it's pretty close to as good as it has been in the MS field with respect to decreasing progression of patients over time. So one of the questions here, and this is speculative, is whether the relatively limited impact on the peripheral immune response, the relapsing remitting biology, and yet as good as we have seen in terms of reducing progression of disability, does this imply that this drug will actually have some important neurobiological effects? It is certainly a very easy to tolerate, very safe medication as it appears through its phase 3 studies, and it may turn out to be a nice add-on therapy in combination to some of the others that are more effective with respect to peripheral immune modulation. And so I'd like to finish up with a few snippets of insight into some depleting therapies in the context of MS, and I'm sure you'll hear more about these in the context of some other of the autoimmune diseases you'll be discussing a little later. This is what happens in multiple sclerosis when you deplete patients in their periphery from B cells using anti-CD20 monoclonal antibody. This happens to be the data set published in the New England Journal a couple of years ago now using rituximab, a chimeric anti-CD20 molecule, and you'll see here, looking again on the y-axis at the development of these new gadolinium-enhancing lesions in the brains of patients, that the placebo group continues to develop these over time, whereas those in whom you've depleted the B cells from the circulation with 2 infusions 2 weeks apart and nothing for the subsequent 6 months have a very substantial decrease in the number of new lesions in the brain. This translated into fewer relapses. This relapse data set comes from ocrelizumab, the more modern humanized version, if you like, of rituximab, and this is showing you against an active control the relapse rate reduction of ocrelizumab compared to placebo. So ocrelizumab at 2 doses here, placebo shown here, and on the right the active comparison with the weekly interferon beta. And so this is to some been a surprise. You remove B cells selectively and you very substantially change the face of this disease. So it is no longer a question of whether B cells are involved, it's really more a question of how are B cells involved. And what's been very interesting is that when you look at the cerebrospinal fluid, and I'll tell you that this drug was introduced into MS because of the notion that B cells may contribute to plasma cells that make bad antibodies, so if we target the B cells, maybe we'll impact autopathogenic antibodies and help patients. It turns out that the spinal fluid, the CSF immunoglobulin and oligoclonal banding pattern, very common in MS, are absolutely not impacted by this treatment in the face of its substantial efficacy. And while you get very profound depletion of B cells in the periphery, you seem to get partial depletion of B cells in the CNS, and interestingly, partial depletion of T cells in the CNS. And this may be because of one of several reasons or a combination. We don't know whether this reflects decreased T cell activation in the periphery. I'll show you some data to suggest that. It may also reflect decreased chemoattraction by B cells within the CNS, so that you decrease the B cells there, there's less T cells coming in. The data that we have generated that suggests that rituximab and anti-CD20 are likely to mediate an important peripheral effect in the context of MS relapses is that when we remove B cells from MS patients, so here's a patient shown pre-rituxan and post-rituxan, the same patient PBMCs that are pulled out and activated in vitro, and then looking at CFSE dilution on the x-axis as proliferation, and this is intracellular cytokine staining, in this case for interferon gamma. So the left upper quadrant would be proliferating interferon gamma producing cells, and here we're getting on CD4 T cells. And you can see in this patient before treatment, if you just focus on the left upper quadrant, a substantially decreased CD4 proliferative interferon gamma producing T cell response. Very surprisingly, at least to me, a substantial decrease also in the CD8 T cells. So both CD4 and CD8 T cells appear to care a lot as to whether B cells are present. And this is also the case, I don't have the time to show you, for decreasing CD4 Th17 responses. So we think, and again because of time I don't have a chance to elaborate on the presumed mechanisms, we think that B cells, in part through the production of pro-inflammatory cytokines, impact the activation of T cells in the periphery. So this is not suddenly a B cell mediated disease. It remains a T cell mediated disease, but B cells seem to play an important role. A word of caution, while people have been depleting B cells, and one of the questions of course is, can we continue to do this for many years in these patients without increasing risks? There's been therefore an attempt to target B cells without killing them, and one example is taking advantage of knowing that there are receptors that are important for survival of cells of the B cell lineage, like TACI, and creating a fusion protein for TACI-Ig, which is one of the receptors for BAFF and APRIL. And what happens is that, in fact, patients with MS who get this therapy get worse. They have more attacks, and atacicept has been stopped in the context of MS while it's being pursued in lupus, as I expect you'll hear. So this is an important reminder to us that just like T cells, B cells are not just bad guys or good guys, and how we modulate their biology can impact the disease in surprising ways. It nonetheless remains attractive for us to think of how we might avoid completely depleting B cells. Can we more selectively target them, mediate the benefit that we've seen as a potential benefit, but with less of a concern over time? And the last therapy that I'd like to introduce is alemtuzumab. It's one of the bigger guns. This is a humanized monoclonal antibody directed against CD52, which is present on many cell types. It has a cytotoxic effect and knocks out many cells for a long time, so you get prolonged effect, and you reconstitute a fairly new lymphocyte pool over many, many months following therapy. This is a therapy that's been approved now for some years in the context of B-cell lymphomas, and shown here now against the high-dose interferon, which is considered to be one of the most effective currently approved therapies as far as the standard immune modulators. This is showing you what happens in terms of cumulative number of relapses, increasing over time fairly rapidly in this active treatment arm with high-dose interferon, and both doses of the drug, with the red being the average of the two, very substantially decrease new relapses in this relapsing remitting patient population compared to active therapy. So this at the moment is looking like the biggest gun and the most effective, short of bone marrow transplantation, in MS patients. However, the price that MS patients might be expected to pay is summarized here, and I'll focus your attention on the lower lines, which reflect the emergence of other forms of autoimmunity, principally thyroid autoimmunity and some idiopathic thrombocytopenic purpura as well. So this is teaching us something important about how this drug works. This has been seen in the context of reconstitution, immunology, and other fields, although this appears to be fairly high and maybe, interestingly, unique to multiple sclerosis, at least in terms of the frequency. So I'm going to stop here. And again, I bombarded you a little bit with an update on our thinking in terms of the complexity and the multiple compartments evolving over time with respect to multiple sclerosis. And we focused on the red circled aspect of the biology here, the relapsing remitting biology, with respect to these therapies. And I will stop here and thank you for your attention.
Thanks. Thanks, Amit. The way we're going to work this, we'll have questions after each speaker, since various people have to run to give other talks at this meeting. And while people are coming to the microphone with their questions, let me just ask you to speculate a bit on the past and future of antigen-specific therapy for MS, because that's something we may not hear much about in other diseases today.
Yeah, so I would have loved to expand on that. It's a great question. So antigen-specific therapy, again, has the attraction. If you knew who the bad guys were, could you just knock them out? The reality is in MS, we don't really know. We think that myelin antigens are relevant, but we also recognize, as Steve Miller had nicely showed in EAE, that there's epitope spread. So the notion that any one antigen is likely to be the bad guy in all patients at all times is almost certainly naive. And this is probably why a couple of these drugs that have been tried in advanced phase clinical trials rather quickly didn't make it. They didn't meet their primary efficacy endpoints. So those drugs like MVP 82 to 98 were sort of dead in the water as far as the MS field is concerned. Bayhill Therapeutics, with its DNA vaccine, has taken a more cautious approach and is pursuing this, as you may know, in diabetes first. But the preliminary data in MS, as a proof of principle, is that you seem to be able to downregulate CD4 T-cell antigen-specific responses to myelin basic protein using an MBP DNA vaccine. So a nice proof of principle in the MS context. What I think we would like to do is to try to understand at any point in time, can we use some kind of an assay in patients, such as these antigen-specific response assays, to profile an individual with respect to who in that individual at that time are the bad T cells? Are they specific to MBP, to PLP, to MOG, or other CNS antigens? And then to introduce to that patient a more individualized antigen-specific therapy, which I think will be the way to go, but we need to understand better how to link the diagnostic and the therapeutic over time. As you might imagine, a major challenge in terms of clinical trial design, regulatory agency perspective, and so on. Sir?
Could you just expand for a minute on what the proposed MOA for laquinimod is?
So the question, for those who may not have heard, what does laquinimod actually do? This is a good question. We don't fully know, but what's emerging now from several, as of yet unpublished animal model studies, and a bit of literature over the years, is that the modulation is occurring to both T cells and B cells, but maybe importantly mediated through myeloid cells. So changing myeloid cell biology in a way that in turn changes the response profile of T cells, including the presumably pathophysiologic T cells.
Yes. Is there a place for combined therapy? As you have shown, the targets are different in each agent, so...
Right. So combination therapy has been a theme that has been very important in the MS world, but challenging. And one of the things that got people scared is when the PML first occurred with Tysabri, it looked as though that this might have been in the context of combination therapy with one of the interferons. We then realized that it's not just that, but there have been other examples like the FTY, the fingolimod, or Gilenya together with steroids. So people have a little bit of cold feet with respect to the combination therapy. The most important combination will likely be the immune-directed and the neurobiological-directed aspect. Now, if a single agent can combine those two biologies, that's great, but it may be that we need to do something in the way of combination, and hopefully, because they're targeting very different biologies, there will also not be an issue of accumulative toxicity. And then the question is, combination, should it be serial, or might you consider a paradigm where you do, should it be concurrent, as people have mostly been trying, or should it be serial, where you give a big hit up front, like an induction type of therapy, and maybe then with a milder, safer agent over time, you can maintain the quiescence of the disease. So it's very much an ongoing story in MS. Scott?
So a question about the data you showed on fumaric acid. Intuitively, that sounds like it would be a ostensibly safe therapy. Could you comment on toxicities that were seen first off? And secondly, the efficacy data you showed for fumaric acid, and this is an impossible question to answer because you can't compare across trials. Where does this fit in the efficacy scale of the things that you showed?
So I love it when someone asks a question and says it's impossible to answer. It makes my job rather easy, right? The safety profile first of fumaric acid. This is a drug where there's been quite an extensive experience with Fumiderm in the psoriatic population in Europe, particularly in Germany. GI upset has been the main issue, which has led some patients to have to stop the therapy. It looks like in MS the twice-a-day dosing is just as good as the 3-times-a-day dosing, which is good news for patients. And BG12, which is not exactly Fumiderm, is a 98% type metabolite, if you like, and is thought to perhaps have a lesser problem with respect to the GI upset. Now, we have seen GI upset in patients. Some had to stop the clinical trial because of it. So I don't think it's going to be devoid of that side effect, but it generally looks otherwise to be quite safe. There's been no issue with opportunistic infections or infections for that matter at all, and multiple systems look to be intact. With respect to the mechanism or the impact, if you like, on where it might fit in, the animal models would have suggested that this would have a mild effect, a modest effect on the peripheral immunology. And in fact, the data that's emerged and presented recently at the Academy and is accepted now in Lancet Neurology looks rather impressive. Now, this is without having head-to-head comparisons, but with the caveat that you cannot compare across trials until you have a head-to-head. This looks to be more promising than we had thought in terms of the impact. So this is perhaps going to be a game changer. When this drug comes out, if there are no surprises, other drugs will have to compete to match it. Yes?
Would you care to comment on the recent approval of the JC virus antibody test in the EU?
Right. So this has been, I think, an important development. And for people who are not familiar, many of us are exposed to JC virus and harbor JC virus, but not all of us. And what seems to make sense is that if you don't have JC virus, then you can't get PML. And that appears to be the case when you look at the data from the trials and from the post-marketing. There are now over 130 patients as of last week who have developed PML in the context of natalizumab or Tysabri therapy in MS. This is less than 1 in 1,000, as I'd mentioned. And none of them who developed PML were negative serologically for anti-JC antibodies. That means that if they didn't have evidence of being exposed, they didn't get the disease. So all of the ones who did get PML had antibodies to JC, and the testing now is being used to try to stratify patients with respect to risk. So if you are tested as antibody negative, it is thought to be pretty good evidence that you have not been exposed to the agent and don't harbor it, and that your risk then is likely to be extremely low. The other that we know is that patients do not seem to get this with up to 12 months of continuous therapy, and so people have been entertaining whether you can do drug holidays, and there's controversy around that we can discuss. But if you only expose patients for 12 consecutive months and then give them a little break, hoping that immune surveillance will return and then redo that, is the second year having the same zero risk as the first year, and can you continue going on like that? So these are some of the important questions with Tysabri.
Thank you for an excellent overview of MS and its therapy. Could you clarify for me a point of confusion? Fingolimod is described as a partial agonist of the S1P receptor, but yet at the same time, I thought one of your slides mentioned that it downregulates the S1P receptor as well. So it appears to be mutually contradictory.
Well, so S1P biology is really very complicated, and this is not just me trying to squirm out of answering the question better. It really is rather complicated. There are multiple receptors. Different individual cells can up and down regulate different receptors at different times. What we learned from doing the neurobiological studies is that even in culture, where you're trying to control things, as you dose in different concentrations of FTY, you have even cycling of receptors. So they come up, they go down, even in the face of the drug there. So it makes for complicated biology. Now we're kind of working our way backwards because we now know the drug is rather effective in patients, and we are trying to figure out which aspects of what it does, when and where, are the ones that are mediating the benefit. But you're absolutely right, this is one of the conundrums.
Thank you.
You show the B-cell depression looks somewhat effective through the maybe through the direct effect depression of the antibody against the BMP around that. That is the one direct evidence. But the another you show the T-cell depression, or the reactivation or depression of the activity through the CD4 or CD8 cells, you said. But do you have some direct evidences, the depression of the antigen-specific T cells against the antigen, that means the antigen, neural antigen or others? Do you have such direct evidences?
So with which therapy? I'm sorry, I just missed the first sentence.
B-cell depletion.
B-cell depletion
yes,
therapy.
Right.
Great question. So yes, there is some evidence that antigen-specific T-cell responses against myelin antigens are diminished. Nancy Munson from UT Southwestern has shown that. And there's a question as to whether B cells that may be specific to myelin antigens themselves are also decreased. However, I don't think there's anything about B-cell depletion in MS that is particularly antigen-specific or relies on antigen specificity. I think what we're learning is that B cells play very important non-antibody-dependent roles in the normal immune system. They can function as APCs, they can regulate responses through cytokines both up and down. They can participate importantly in lymphoneogenesis and trafficking of cells. And so when you remove them, you take away their capacity to do that. And even if they are not primarily a problem in MS, which, by the way, the jury is out, they may still turn out to be, but even if they are not primarily the problem, taking them away will take the edge off of T-cell activation in anybody who has B cells depleted, not just in MS patients. And I think you may be hearing about that in subsequent talks. It turns out that in MS it translates rather surprisingly to a very substantial decrease in new relapsing biology.
One other question. Have you ever tried the anti-T cells such as against the CTLA-4 antibodies?
CD4?
CTLA-4.
CTLA-4.
Yes, right. Against the T cells. You haven't tried to...
So in the MS field, there have been treatments with CTLA-4 Ig.
Yeah.
This, Samia Khoury, particularly out of the Brigham and Women's Hospital, has been championing these. And of course, to those who may not be familiar, this is targeting a T-cell co-stimulatory pathway, the B7 co-stimulatory pathway. And it's attractive to try to initiate or to induce responses through CTLA-4, which is thought to be a negative or a down regulator of T-cell responses. The data in MS has reached just beyond phase 1 into phase 2 clinical trials, and there is some promise of efficacy, but they have not really gone through in terms of enough of an exposure to really comment about safety as well as efficacy in more advanced studies.
Thank you.
Thank you.
Thanks for the talk. In immunotherapeutic trials of MS, is there, when patients are enrolled, in terms of their kind of current state of disease, in terms of previous numbers of relapses, that's controlled for? And do you see with these different immunotherapeutic agents differential risk reduction in the different groups depending on where you are in the progress of your disease?
So if I understand, you're asking whether we consider applying different therapies to different parts of the MS spectrum. Is that the question?
Correct. And do you see differences in risk reduction with the immunotherapeutic agents depending on your stage of disease?
So one can say as a generality that as far as immune-targeted therapies in MS, if you remember the little cartoon of the blue and the red biologies over time, over the years, new therapies were initially introduced into the population of patients who were already pretty bad, and it seemed to be reasonable to try to expose them to experimental therapies. What we've learned is that immune-directed therapies work less well in that later stage of the disease and work better in terms of the magnitude of the impact on decreasing new relapsing biology the earlier that you start, even at the time of the first clinical attack that we then realize is the first one of multiple sclerosis. So there's a strong drive in the MS field to try to make the diagnosis earlier than ever before and to try to provide patients with an opportunity to have these therapies decrease new relapsing disease activity with the built-in premise that decreasing that form of injury early on may translate into changing the trajectory of progression later on. We don't actually know that for sure yet.